In this lecture, we are going to go through the whole process of humoral immunity to the detail that I require for my course. So, the goal of humoral immunity, as stated in the prior lecture, is to create antibodies. And antibodies are magical molecules because they make phagocytosis so much easier. They allow for agglutination, and they also prevent attachment for toxins and viruses and even some bacteria. In this example, we are going to use a triangle to represent the antigen that we are trying to combat with humoral immunity. And the goal of this lecture is to really show you the steps involved that end or culminate in creating these antibodies. It's not a short process. There's actually quite a few players involved in this complex pathway. So let me start by introducing the 2 or 3 major players that are going to play a role here. And we are going to start with one that we saw in the prior lecture, and this is a macrophage. So macrophages, and this is a macrophage here on the screen, so I know it kind of looks like an octopus, but it is a macrophage. Macrophages have the ability to phagocytose many pieces of debris or particles or even bacteria. And here you can see an impressive picture of a macrophage sending out its cytoplasmic extension. So there's a cytoplasmic extension, and it's wrapping it around a bacterial cell. And basically what it's going to do is it's going to bring that bacterial cell in and digest it. And when it digests it, it's going to pick the antigenic fragments off the surface and display them in MHC II. So our story is definitely going to involve macrophages that are going to become what are called antigen-presenting cells. So we've already learned some about macrophages and their ability to digest antigens and present them on the surface of their cells in a prior lecture. Now we are going to look at some new players that we have not yet really explored. And these are going to be lymphocytes. So lymphocytes actually come in several different subcategories. And one subcategory is called a B cell. And B cells are produced and mature in the bone marrow, the red bone marrow specifically. And B stands for bone. The other category of lymphocytes that we're going to look at are called T cells. And T cells mature in the thymus. So T cells are named because these mature in the thymus, which is actually an organ that is part of the lymphatic system. Okay, T cells actually come in several subcategories. And you can name T cells based on what types of receptors they express on their surface. So one way that you might read about T cells is called CD, almost like a compact disc. You don't need to write this down, but I'm just going to list it here. And so CD stands for cluster of differentiation, which is a really strange word for a receptor, but that's what it means. And CD, there are several subcategories of cluster of differentiation receptors, and one of which is 4 and one of which is 8. So if you ever go into the literature and read about T cells, you will often see T cells referred to as CD4 positive. That means that they carry and express cluster of differentiation 4, which is just a certain type of receptor, or CD8 positive, which also expresses a slightly different type of receptor. While this is interesting, it doesn't really tell us too much about what that means. So if a T cell expresses CD4 or CD4 positive, what does that mean? If it expresses CD8, what does that mean? So we're not going to worry about this. Instead, we're going to classify T cells based on their function. And while there are more subcategories than I'm listing here, the two major subcategories that are appropriate for our level of immunology are called helper T cells and cytotoxic T cells. So helper T cells get the abbreviation T with a subscript H. Interestingly enough, helper T cells are most often CD4 positive. So again, if you go into the literature and you see CD4 positive, it's probably a helper T cell. The other one, cytotoxic T cells, do not play a role in humoral immunity and therefore will not be included in this lecture. We'll talk about them in the next lecture when we do cell-mediated immunity. But for now, we're just looking at helper T cells. Okay, so now that we've kind of introduced some of the players, let's turn and take a look at what happened at the end of inflammation. So at the end of the inflammatory response, a monocyte is compelled to emerge from the blood and through cytokine permission turns into an active phagocyte, and that active phagocyte is known as a macrophage. So macrophages tend to have cytoplasm that looks a little bit more like an amoeba or a sea star rather than a sphere. So the activated macrophage phagocytoses this bacterium, and the red triangle is the antigen on the surface. So I'm going to go ahead and do a quick drawing to show you how this activated macrophage phagocytoses and then takes the antigens and displays them on the MHC case. So the first thing we're going to do is engulf this bacterium in its own vesicle. So this is ingestion. Now, phagocytosis is really a two-part process. Ingestion is the first part. Now we need to digest the bacteria to destroy it. If you don't destroy the bacterium, then you haven't really gotten rid of the problem. And there are certain microbes out there that are really good at actually allowing themselves to be ingested, but preventing the digestion. And tuberculosis is one of them. So tuberculosis is a bacterium that actually allows the macrophages to go ahead and ingest them, but then they prevent the digestive part of the phagocytosis. And so the macrophage never kills the tuberculosis bacterium, and then the tuberculosis bacterium replicates, and then you're infected. So it's really kind of a clever defense by the bacterium to prevent digestion. And I only mention that so you understand that both steps are necessary for phagocytosis to work. Let's assume this is not tuberculosis and we successfully digest it. The way that we digest it is by taking a lysosome, which remember is a vesicle containing digestive enzymes, and then we fuse it with this vesicle, and then these digestive enzymes come out and basically start eating this guy up. But we're not going to digest all the parts. So some of these fragments are still going to stay. Some of them will probably be digested, and then most of the bacterium will also be digested. Okay, so once we do that, then the cell has a way of taking the antigenic fragments and inserting them into these special display cases called MHC. And MHC stands for Major Histocompatibility Complex. And there are two MHC varieties that will be important to our immunology story, and they are MHC I and MHC II. I don't know for sure, but I would suspect that this is the order in which they were discovered. Macrophages are going to use MHC II. So MHC II are these display cases that you see here in blue. Now MHC II is something of a trophy case for these antigens. And the way I think of this is very similar to the Predator movie and franchise. So if you're familiar with Predator and the franchise Predator, it's based on an alien species that goes around the galaxy and kills for sport, and then collect the skulls and display them as trophies. So in the same way, I feel like the macrophage is a predator that eats up these microbes and then wants to brag about these antigens that they've collected. So once they have an antigen in a MHC II case, then they go ahead and put that on the surface like so. So at this point, we would say the macrophage is antigen presenting for the triangle pathogen. Now, one thing that's interesting about macrophages is that they can present many different antigens. And once a macrophage starts presenting antigens in MHC II, again, we would call that an antigen presenting cell. So we use the abbreviation APC. So it is an activated macrophage, but it is also an APC macrophage. And I will use that terminology quite a bit on the test. So an APC, an antigen-presenting cell, is any cell in the body that is capable of presenting antigens in MHC II. So this is an important definition. So it's important that you understand that it has to be MHC II. If you use the prefix APC, you are specifically saying MHC II. It cannot be MHC I. And MHC I does not play a role here in humoral immunity. It does play a role in cell-mediated immunity. And although we will not get into it, it also plays a role in cancer. MHC II is a trophy case, and an immune cell that is conquered, it's phagocytosed, it's digested, and it's displaying the antigen as a trophy, that is an antigen-presenting cell. So which immune cells are capable of being antigen-presenting? Is it all of them? No. Macrophages, B cells, and another type of cell that we're not covering called a dendritic cell are the only antigen-presenting cells in the body. So for our purposes, it's just macrophages and B cells. Okay, let's assume that this macrophage has gone out and collected a bunch of other antigens from other conquests and is now showing off all of its trophies. So what happens to this macrophage? What does it do? The next thing the macrophage does is it enters the lymphatic system. So at this point, we're going to take a break from our discussion of the macrophage and start looking at the lymphatic system. The lymphatic system is a collection of lymph nodes, lymph vessels, and a few select lymph organs that are running kind of parallel to the circulatory system. I'm not going to go into too much detail here, just enough detail for us to understand the physiology. But I want to remind you that when you did the cardiovascular system lectures, you learned, and this is a capillary, you learned that the pressure in the proximal part of the capillary was greater than the interstitial fluid, And so fluid has a tendency to move out. And then as blood moves downstream towards the distal end of the capillary, the blood pressure drops, and that tends to draw fluid back in. But not all of the fluid goes back into the distal part of the capillary. Some of the fluid goes instead into the lymph system. So the lymphatic system has a really low pressure. And off the top of my head, I can't remember what it is, but I want to say it's like 15 millimeters of mercury. It's something really low, lower than any capillaries. So it acts like a weak suction. Think of like, a kind of a cruddy dust buster or something where it's going. So weak suction. And so it's always sucking some fluid in. And you'll notice something about these lymph vessels is that they have these kind of terminal ends. So what they're going to do is they're going to collect fluid from all of this interstitial fluid here. And then that fluid is going to start moving away from these terminal beds towards a bigger vessel. And I'll show you that in a moment. The lymphatic system runs in parallel to the circulatory system for most of the organs. And when you have some sort of inflammatory event, remember one of the consequences of inflammation is that you're going to get a lot more fluid exiting the capillaries because the capillaries get really leaky. So what that does is that causes fluid accumulation here in the interstitial fluid. Now the lymph should be draining that, but if the lymph cannot drain it faster than the fluid is leaking out of the capillaries, then the fluid accumulates here, and that's edema. So edema usually will eventually subside as the lymph system kind of catches up and takes care of the extra fluid. But what happens if your lymph system is compromised or some of it is removed? Then it's very difficult to deal with the accumulated ISF fluid. And we see that sometimes. We see people who have had part of their lymph system removed have recurrent swelling in the same limb where their lymphatic system is removed or their lymphatic system is blocked. And again, that underscores the importance of the lymphatic system in draining fluid from the interstitial fluid. So again, not all the fluid goes back into the capillaries. Some of it goes into the lymph. So if we take a look at the lymphatic system, you can see it really is a network of vessels, not terribly dissimilar to the network of blood vessels. It's different for sure, but it kind of runs all over the body. And so when you take a look at a lymph vessel, you can envision that lymph, and that's what the fluid is called there, is going to move through these lymph vessels and then eventually hit these kind of nodes. And those are called lymph nodes. And if you look to see like where there's aggregations of lymph nodes, you'll actually see kind of three big aggregations. This picture doesn't show it real well, but there's a huge aggregation around the groin. There's a huge aggregation in the armpit, and then there's a huge aggregation around your neck. These serve as checkpoints of sort for lymph, basically returning from a limb, your arms, your legs, or even your head, before going back to the core of your body, the trunk. And that is because once lymph passes through those lymph nodes in the axillary region, the groin, the neck, then that lymph is going to get returned into the blood. So in this way, the lymph nodes act like a cleansing station where debris and pathogens are removed. And the lymphatic system plays a role in the immune response, as we'll see here. Let's return to our macrophage and take a look at how the macrophage is interacting with the lymphatic system. So once the monocyte comes out of the blood, becomes activated as a macrophage and consumes the bacteria or whatever other debris might be there and becomes antigen presenting, then the next thing that's going to happen is that macrophage is going to get sucked up into the lymphatic system. And from there, it's going to move up the lymphatic system. Now, what's it doing once it moves up it to the lymphatic system? The next thing it's going to do is it's going to run into the lymph node. And so remember, the lymph node has a large number of lymphocytes, including B and T cells stationed there. And so the macrophage is going to come in through one of these vessels and basically start trying to talk to specific lymphocytes. Now we're going to take a look at some of the lymphocytes and how they might play a role here in humoral immunity. So far in our story, we have a macrophage becomes antigen presenting for the triangle antigen after phagocytosing the bacterium. The antigen-presenting macrophage gets sucked up into the lymphatic system, and now it's basically visiting lymph node and lymph node and lymph node. So it visits lymph nodes in succession, and at each lymph node, it's going to be talking to specific lymphocytes. Now I'm going to stop the story and kind of jump ahead a little bit. Remember, our end game is to make antibodies. So who makes antibodies? B cells. So B cells are lymphocytes that make antibodies. Each B cell can make one and only one antibody. And so you can see here I have 6 different B cells, and they all make slightly different antibody shapes. We call these different B cells B cell clones. So while I have 6 displayed here, you probably have thousands of different B cell clones in your body. And we're looking for the right B cell, the B cell that has the antibody that's going to match the antigen that's causing us problems. For right now, that antigen is a triangle. So can you tell which one of these B cells, and I'll just go ahead and number them. These are the clone numbers. So which B cell clone number is going to match the triangle? That one. So that's the only one that's going to match a triangle. In reality, antigens are much more complex than geometric shapes. Antigens act more like keys. Think of like house keys or office keys or building keys. There's a lot of differentiation in the ridging. So if I made a key that looks like this, then in terms of making a lock, I would need a lock that fits exactly that shape. If I change the shape a little bit, this second iteration here is different enough that it will no longer open the same lock. So think of antibodies like keys. So think of antigens like keys, whereas antibodies are like their complementary lock. And that's the analogy that you want to think of when you think of the complexity of antigens and antibodies. Our bone marrow makes thousands of different B cells. Each B cell makes a single antibody cutout shape, a single lock, and it's random. It's not designed for a particular antigen. The analogy is that you are a locksmith and you're basically told make a lock. And you say, well, what key am I making this lock for? And the customer says, I don't know, just make a random lock. And you make a random lock. Is there a key out there somewhere in the world that will fit your lock? Maybe, maybe not. So the body does not know which antigens it will ever encounter in your life. So what it does instead is it tries to diversify its B cell portfolio by making as many different locks as possible, so that when presented with one random key, you hope to goodness that there is an antibody out there that's going to match it pretty closely. The only way that you can hedge your bet, so to speak, is to make lots and lots and lots and lots and lots and lots and lots of different antibodies. So this is a really interesting strategy. The body diversifies its portfolio of B cells. It never knows which antigen it's going to encounter, so it tries to make as many different types of antibodies as possible. The only criteria is that the antibody cannot bind to you. Will it bind to anything actually produced by a pathogen? We have no idea. We just hope that, yes, that when you are exposed to a pathogen, somewhere in your body is a B cell that has an antibody that matches. That's what you hope. There are cases where people routinely get vaccinated to a particular antigen and they never develop antibodies, suggesting that there are in fact cases where you don't have an antibody that matches that antigen at all, anywhere in your body. So it's not a perfect system. And it's important to understand that this is not design on demand. I think many people, when they try to envision immunology, believe that B cells are a design on demand type of cell. That is, they take an antigen and then they're like, oh, here's a key, let me design a lock to match that key. That is not what happens. So be clear. Just because I show you an antigen doesn't mean that a B cell is going to be able to make an antibody that matches it. Instead, you make a whole bunch of different antibodies in advance and hope that when you actually get that antigen, somewhere in your body is this B cell. Now, where do these B cells like to hang out? The lymph nodes, of course. That's their kind of favorite place of residence. It's not the only place that you're going to find them, but that's where they're very common. So, if we have this triangle antigen, then we know that the B cell that we're going to want to basically become active and make a whole bunch of antibodies for us, because when that B cell starts making their own antibodies, they make identical copies of what they display, is going to be this one. Okay, so we call that process of identifying the lymphocyte that has the matching, in this case, the matching antibody, the complementary antibody, we call that clonal selection. So in clonal selection, we're basically identifying B cell 832 as our winning B cell. And so now what's going to happen? Once that B cell is activated, it is actually going to go through a series of cell divisions. Now, I don't want you to draw anything out yet, because the way that I've drawn this isn't great. It's not perfectly accurate. But we're going to go ahead and say, hey, I want to make a bunch of you. And so it's going to go through its own version of cell division. And then what these cells are going to do is they're going to make and secrete a whole bunch of the identical antibodies. How many antibodies can be secreted? Like 2,000 per second. Huge numbers get dumped into the bloodstream. Now notice that they can only make the exact same antibody that they advertise. They can't make one with a different variable shape to it. They can't make a square. They can't make a semicircle. They can only make the ones that fit the triangle. So this is our end game. And again, don't draw this out because I drew it out incorrectly. I'll draw a more appropriate one in a little bit. This is our end game. Our end game is to identify the right B cell, activate it and say, hey, you're the winner. Go ahead and reproduce and have your daughter cells, which are just the cells that come from that cellular division, make and secrete a whole bunch of these antibodies into our bloodstream. And they'll go into the bloodstreams, the interstitial fluid. They'll basically go everywhere in the extracellular fluid. And then we know what's going to happen. We know that they're going to bind to antigen they encounter in the extracellular fluid. It will make phagocytosis easier. It will prevent binding. It will cause agglutination, which also makes phagocytosis a whole lot easier. And then the tables turn. Now you are winning against this pathogen because you are phagocytosing them faster than they can replicate. So this is our end game. So now that we've looked at our end game, find the right B cell, get the B cell to replicate. Each B cell then should be making a whole bunch of antibodies. We're going to dump them into the bloodstream and we win. The real question is, how do we go from the macrophage that was antigen presenting in the first place to this clonal selection of B cells? And this is where things get a little bit hairy because it's not clean. Now, what you would probably expect is that macrophage should be able to talk to that B cell directly. And this seems like a reasonable, don't write any of this down by the way, because it's wrong, but this kind of seems like it should be reasonable, right? I should be able to like maybe match that B cell antibody to that antigen and go, yes, we're the winner. I mean, that seems reasonable. Like I have an antigen presenting macrophage. Right here, I have that triangle antigen. I'm showing it off. I go into a lymph node. I call over this B cell that has, and I basically ask the B cells to try out their antibodies on the various shapes that I'm displaying. And when I get binding, that cell-to-cell contact, that's a type of communication. And you'd think, yes, this is what activates the B cell. We're good. Wrong. If only it were that simple. Here's the problem. Antibodies can only bind to what is called free antigen. This is really important. So what I just did does not actually happen. So what is the definition of a free antigen? Well, a free antigen is one that is not in an MHC case. So is this that I've circled a free antigen? No. Why? Because it's in an MHC case, so it's not free. So as a consequence, that antibody cannot bind. And so there is no way for the macrophage to talk to the B cells. Let me ask you another question. Is this a free antigen if you just saw that all by itself? Yes, that's a free antigen. And there are antigens that are secreted from bacteria like toxins, and they would just be kind of free floating. That would be a free antigen that could bind to the antibody displayed by the B cell. What about this? Are these free antigens? Surprisingly, the answer is yes, they are. Because by definition, they are not in an MHC case. You might say, but they're attached to bacteria. That's fine. They're not in an MHC case. So our antibodies are not allowed to bind to anything in an MHC case. The MHC case prohibits it. But if it's not in an MHC case, it doesn't matter where else it could be. It can be antigenic and it can bind to an antibody. Let's now address this issue of how can we get the macrophage that was kind of the first responder, phagocytoses the bacteria, and presents that antigen on its MHC II case. How can we get it to actually say, hey, B cell, you're it. You're the one I want. I want you to go through clonal selection, when they can't actually talk to each other. And the answer is we need a middleman of sorts. We need a liaison. and the liaison is the helper T cell. The macrophage is going to go from lymph node to lymph node, and every time it goes into a lymph node, it's not going to want to talk to a B cell. It's going to ignore all of the B cells. Instead, it's going to want to talk to a helper T cell. Now, this is where it gets really interesting. Helper T cells, and I have 3 drawn down here, each carry a specific receptor. And the receptors are called T cell receptors, and I have them drawn in green. Now, the T cell receptors also vary in their cutout shape. And just like B cells, these cutout shapes are random. And guess what is supposed to fit in that cutout shape? A potential antigen. But what's different about T cell receptors, and these are special proteins expressed by T cells, is that the T cell receptors will only bind to the matching antigen in MHC. So whereas B cells cannot bind to antigen in an MHC case, T cell receptors can only bind to antigen in an MHC case. And here I have a B cell, and don't worry about how I got this yet. I have a B cell that is presenting, it's an antigen-presenting cell, presenting the triangle antigen in an MHC II case. And so you can see here, no match, no match, match. So notice that they fit together like puzzle pieces. So the T cell receptor has a cutout, and each cutout is different, and each cutout is meant to bind to a particular antigen. Just like B cells, these cutouts are made randomly and in advance. And so once again, we have different clones. And the body aims to diversify its portfolio, and you have thousands of different T cells out there, each with their own T cell cutout. So let's go ahead and give these clones some names. And in our case, T helper 9815 is going to be the winning T cell here. T cell receptors only bind to complementary antigen in MHC cases. T cell receptors can never bind to free antigen. You might notice that I kind of put these little tiny hooks on the MHC case. Those hooks grab onto the T cell receptors. So when I go ahead and try getting this guy to bind here, those hooks basically grab on, and so they grab on here. And that grabbing is the cell-to-cell contact that basically stimulates conversation between these two cells. So once that grabbing occurs and the fit happens like a puzzle piece, whatever that antigen presenting cell is on the left is now talking to that T cell, and they're having a conversation. Okay, so I'm basically laying out different parts of the story without giving you the full story. So we're doing this kind of in a piecemeal fashion, but now we're going to try putting it together. Okay, so let's go ahead and revisit our story. An antigen-presenting macrophage walks into a lymph node and calls over all of the helper T cells, and there might be a couple hundred in there. And it says, hey, let's play touchy-feely. That's their version of show and tell. And they basically say, let's see if we can try you out. Do you fit? No, you don't fit there. Do you fit here? No, you don't fit there either. Do you fit here? No, you don't fit there. Okay, thank you. Next. And they basically audition. They'll audition every T cell until one fits. Now in our case, the one that we're looking for is going to be T helper 9815. And so it's going to basically take 9815 and it's going to bind. And when these link up and attach like this and they go, yes, yes, we have a good fit, the macrophage is going to secrete cytokines. So this is my cytokine. It's like fairy dust. And it's a paracrine. Cytokines are a paracrine. But the only cell that is responsive to those cytokines are going to be whatever cell is attached to that macrophage. So while this other T cell might be in the line of fire of those cytokines, it does not do anything. It does not have a response because it's not attached. So the only cell that is going to respond here is going to be T helper 9815. So what that does is that activates the attached helper T cell. And what does activation do? Well, this is kind of like a form of clonal selection. So what's going to happen is that helper T cell is now going to replicate. And each daughter cell is going to be active and is going to go in search of a B cell. So the middleman or the liaison is the helper T cell. So the chain of events is this. The macrophage is going to talk to, through cell-to-cell contact, the helper T cell, who's then going to go and try to talk to the B cell. And again, the goal is to identify the correct B cell and initiate clonal selection. So the macrophage never directly talks to a B cell. Okay, so this helper T cell is now going to replicate itself and produce a bunch of daughter cells. And each one of these is going to be active. And this macrophage is going to go on its merry way. It's going to keep going through the lymphatic system somewhere else and try to find additional T cells that it can activate. So meanwhile, these helper T cells are now going to disperse. They're active. They're like primed and ready to go. So I'll put a little like, they are on fire, ready to activate a B cell. And they're going to go around and try to find the right B cell. Well, we know what the right B cell is, right? We already saw the right B cell. So let's say that this helper T cell gets in proximity with this B cell. And you know that this is the right B cell. That these guys should match, right? So how are they going to match? I don't know. See, we can look at these and go, yes, these are matching. They have the same cutout. But they don't have eyes. They can only do touchy-feely. So there's no way for these guys to touchy-feel and go, hey, do you have the same, complementary cutout that I have? I don't know. So here's where things get a little interesting. The B cell, when it's advertising its antibodies, as shown here on the left, cannot interact with the T cell in this form. We call a B cell where it's advertising its antibodies a naive B cell. In fact, a naive lymphocyte of any type is one that has never seen its antigen. So in fact, these helper T cells, when they're activated, because they were able to bind to the antigen in the macrophage, they're no longer naive. This is not a naive T cell. But this one is. Okay, So going back to our naive B cell, a naive B cell cannot be activated. It cannot go through clonal selection. Instead, the naive B cell must have seen the antigen itself. So how does this work? If you truly have an infection, then some of these pathogens also get sucked up into the lymphatic system. And when they get sucked up into the lymphatic system, they bounce around and they run into things. So they're bouncing around. They're not going to bind here. Why are they not going to bind here? Because a T cell receptor cannot bind to free antigen. So they're not going to even bind here, but they might bounce around and bind to the antibody. So in this way, antibodies are like lures and the B cells are fishing. And so when they catch the antigen that matches their lure and it gets hooked like this, you know what happens? They basically invaginate, bring whatever's attached into the cell, and then they pluck off what they have captured and then put it on an MHC II case. So they become antigen presenting. So this is an antigen presenting B cell. And once it's an antigen presenting form, it no longer displays its antibodies. It doesn't need to. It fished out what it wanted. So the helper T cells, when they go from lymph node to lymph node looking for the B cells, are going to ignore all the naive ones and are only going to want to interact with antigen presenting ones. So now what we have is we have a B cell that is activated by a helper T cell. So we have this cell-to-cell contact once again. And so what we're going to do is we're going to see cytokine, fairy dust, being secreted here, fairy dust. And that's going to activate this B cell. And what does activation means? It means make a bunch of daughter cells, and those daughter cells should be secreting antibodies. Okay, so now we've got several of the players, or this kind of the stories, the fragments of the story laid out. We're going to try putting the whole thing together into kind of one cohesive linear story, because we've done this kind of piecemeal. Okay, so our story starts with an antigen presenting macrophage that is going to enter the lymphatic system and move up to the lymph nodes. So you can see here that in the lymph nodes, I have two different cell types. On the right, I have B cells that are naive, that are displaying their antibodies, and on the left, I have T cells. So basically, this macrophage is going to go in, and it's only going to try talking to these T cells. Okay, so what does that look like? That looks something kind of like this, where this macrophage comes and basically says, hey, helper T cells, come over here, try your stuff out. Nope. And we have a winner. So now that we have a winner, we'll go ahead and secrete cytokines. That's going to cause a bunch of these duplicates. They're going to create daughter cells. And then each of these are going to further disperse into the lymphatic system. So now these helper T cells are going to move from lymph node to lymph node. And so what they're going to do is now whenever they go into a lymph node, they're going to call over only antigen presenting B cells, of which there are probably not that many. So if we come back here and we look, we can see that there is an antigen presenting B cell. It's right there, but there's not that many relative to the number of naive B cells. When the helper T cell goes into the lymph node, it cannot bind to any of these guys. So it's only going to talk to antigen presenting B cells. So in order to go through clonal selection then, that B cell, that naive B cell that's shown here, had to bind its antigen and turn into an antigen presenting B cell. And once that happens, Then this helper T cell can go ahead and bind. And you know what? Once that helper T cell binds, it secretes cytokines. The cytokines activate that B cell. And now this B cell is going to go through clonal selection. So only APC B cells can be activated for clonal selection. Naive B cells cannot be activated. So what does clonal selection actually look like? Because when I drew it before, I still had the antibodies displayed. And it turns out that when this makes a bunch of daughter cells, most of the daughter cells are going to morph into what are called plasma cells. So this is kind of a new piece of information here. Plasma cells have a lot of endoplasmic reticulum to them because they make huge amounts of protein. So in fact, if you have my workbook, on the very back, there's a watercolor drawing of a plasma cell, and it's all endoplasmic reticulum. And so we're going to go ahead and make thousands of these. So most of the daughter cells differentiate into what are called plasma cells. And plasma cells are the ones that make the antibodies. They're short-lived. They don't live very long. And these guys are going to mass secrete huge quantities of antibodies. Something like that. Okay, so plasma cells are the antibody secreting factory. They themselves are no longer displaying their antibodies, and they're not really doing anything except making and secreting a whole bunch of antibodies into the bloodstream, which is then going to go to all the extracellular tissues. But some of the daughter cells do not differentiate into plasma cells, but instead return to their original naive advertisement state. In doing so, they regain their naive state. So what that means is that we now have more copies of the original clone in the naive state, and they're not active, and they can be reactivated by subsequent exposure to antigen, but we have more of them than what we had initially. And these get reshuffled throughout the lymphatic system, and we have a special name for these. These are called memory cells. And we're going to look at the function of memory cells when we get to lecture 5. Okay, so that finally concludes this very long lecture. Thanks for sticking with me, and hope you were able to follow the story.